Hello friends, Ace Botany, next episode. In this episode, we'll be discussing a new chapter, that is chapter number 10 from NCERT, Cell Cycle and Cell Division. A small chapter, normally 3 episodes sufficient. The exam point of view, either theory or entrance, it is important. Roughly 3 questions for NEET exam and some theory questions will be there for theory exam, including practicals. So we can proceed. So first I will share the screen so it will be convenient for you. I think screen came, so we can proceed. So what all items will be discussed in this chapter? For that, NCERT content is given here. And in this episode, what we will be discussing, that is given here. So we can proceed. First, we can start with the term introduction. In cell, the basic unit of life, you may be studied a item from schools or a +1, +2 level, a statement called omnicellula is cellular. What it means? A cell arises from pre-existing cell. So just like a line, the theory is otherwise called cell lineage, and scientist is Rudolf Virchow. And that itself indicates what is the need for cell division. That is, a single cell, from a single cell, like zygote, an entire organism will be developing, that is through cell division. So now we got the importance of cell division, and this chapter mainly deals with the cell cycle, different types and importance of it. I think introduction is clear, generally for the chapter. Now comes the term cell cycle. When we study cell cycle, 2 basic terms we will remember. One is haploid cell N, other is diploid cell 2N. For human beings, haploid cell N equals 23, that is in gametes, while 2N is diploid, that is 46 chromosomes in the cells. So once again, haploid 23 in human beings in gametes, N, and diploid 2N, that is 46 in body cells. I think idea clear. And what is the meaning of the term cell cycle? Cycle means something that will be repeating in a regular interval of time in a regular fashion. Here also almost same meaning, but concerned with the cell division. So sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into 2 daughter cells, termed as cell cycle. This is the classical definition given in NCERT page number 120, March 24 reprinted edition. And who will control cell cycle? Cell cycle is basically genetically controlled event, genetically controlled. And what are the reasons for cell division, or why a cell divides? What makes a cell to divide? Different reasons are there. Some of them include to maintain surface area volume ratio, to maintain nucleocytoplasmic ratio, which is otherwise called Kern plasma ratio. Then certain substances will be there that will enhance cell division. Similarly, from a single cell, it has to develop into an organism. Specifically, multicell organism, it comes, cell has to divide. So these all are some events by which makes a cell to divide. Now comes mitogens and mitotic poison. GEN, gen normally means to produce, to develop. So mitogen means substances that will cause mitosis or cell to divide. Plant side and animal side, many substances are there. Plant side, hormones like cytokinin, auxin, gibberellins. Animal side, EGF, epidermal growth factor, here I have written here. PDGF, platelet-derived growth factor, and insulin. These are some mitogens. So practically, antagonistic way, what are mitotic poison substances that will prevent the cell division at various stages, and which include colchicine, from a plant Colchicum autumnale, then mustard gas, here you can find, then chavolans, azides, cyanides, ribonucleases, etc. Of which, exam point of view, predominant one is colchicine. Now comes the term checkpoints. So wherever the term checkpoints comes, what is the meaning? Checkpoints are the control mechanism which ensures the events are correctly taking place in a proper order in any place. That is discipline. Here checkpoint means all this concerned with the cell division. And when the decision comes that a cell has to divide, answer for this question is in a phase called G1 phase. And basically, two checkpoints will be available. One is from G1 to S, and that phase is the major checkpoint. Here you can find. And this is otherwise called restriction point. Second is between G2 and M. That is minor checkpoint. It is linked with the protein, and this procedure is concerned with enzymes, proteins, chemicals, energy, many are involved. When once G1 to S is crossed, normally cell will divide. I think that idea is clear. Checkpoints, again, exam point of view, important. Now, cell cycle, different phases, basically two phases, I phase and M phase. Here written, I phase. What is the full form? What is the meaning? What all events will take place, time, etc. So when we come to I phase, interphase, preparatory phase, resting phase, most active phase, non-dividing phase, biosynthetic phase, organelles and DNA will be replicating. And I phase consume more than 95% of the entire cell cycle. NCERT given a data in humans 24 hours needed for the completion of cell cycle. Similarly, yeast cells need 90 minutes. So respectively 24 hours or 90 minutes, it's more than 95% will be consumed for I phase. Problem type question. Now comes M phase, mitotic phase. Cell division takes place and it is commonly called most dramatic phase, and this is famous NCERT diagram for labeling, etc. Now we have to proceed to interphase. Interphase broadly divided into three, G1, S, G2. Three phases, G1, S, G2. And first phase is G1 phase. Many synonyms will be there. Growth phase 1, gap phase 1, post-mitotic phase, or pre-synthetic phase. Before and after and before, that way. And here, no synthesis of DNA. Size of the cell keep on increasing. Chemicals like ATP, RNA, protein, amino acid, nucleic all will be synthesized. And most of the cell organ duplicates in G1 phase. And the duration of G1 phase differs. How the difference comes. See, if the cell divides frequently, G1 will be shorter. Otherwise, it will be longer. And when we compare G1 as G2, normally G1 is the longest among S and G2 when we compare. G1 phase can have three fate or three directions. One, it will continue a cell cycle and enter into a space that way it proceeds. Two, stops at G1 phase, proceed to G0, then undergo differentiation. Three, get arrested in G1 phase, then enters into G0 phase or come back to cell cycle and continue. So three fates will be there. And these all events connected with the mitogens, energy, checkpoints, etc. Normally, G1 will be greater than S, S will be greater than G2. So in 24-hour duration, we can find roughly G1 is around 10 to 11 hours, S will be around 8 to 9 hours, G2 will be around 4 to 5 hours. It is variable, still normally G1 takes maximum time among interphase. G1 phase is most variable period because due to the physiology, condition, environment, everything may last for days, months, even years. Now comes G0 phase. So in this phase, cell cycle will be inactivated G0. It may be due to lack of mitogens and energy-rich compounds. So no stimulation. Cells in this stage remain metabolically active, but no longer proliferate unless called on to do depending on the requirements of the organism. This is a famous line from NCERT page number 122. Cells of the quiescent stage do not divide further. In that stage, they cannot divide. Exit from G1 phase and enters into quiescent stage, G0. Now, example for G0 phase cells, heart cells, nervous cell, that is why they are, when a wound comes, not easy to repair. These cells may act as reserve cell. From plant side, that is in connection with the procedures like differentiation, de-differentiation, re-differentiation, or even connected with the secondary thickening, we can find this G0 phase. Again, famous NCERT diagram. Now comes S phase. S phase or synthetic phase or post G1, pre-G2. Invisible phase of cell cycle, scientists have no idea what is originally happening previously. Chromosome replicates, DNA content doubles. S phase or synthesis phase marks the period during which DNA synthesis replication takes place. In the initial amount of DNA, if it is denoted as 2C from NCERT, then it increases to 4C. Remember, no increase in chromosome number. Chromosome number will be same. No increase in chromosome number, DNA get double. In animal cell, during S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. Animal cell. That is why centriole. And events will be like doubling of DNA, histone production, no centrioles, all will be in S phase. Then comes next phase, it is called G2 phase, gap phase 2, growth phase 2, post-synthetic phase, pre-mitotic phase. Cell growth will be continuing, DNA synthesis stops, protein RNA synthesis continue. And in G2 specifically, we can find tubulin synthesis, cell organs like mitochondria, chloroplast, Golgi body duplicates. Remember, most of the cell organ duplicates in G1 phase, but mitochondria, chloroplast, Golgi body duplicates in G2 phase and cell gradually prepares for division. After that, 5 theory question, 5 NEET model question with the key. So what all things we learned in this chapter? Just a rewind. That is cell cycle and cell division, chapter number 10 of NCERT. Here, content of the chapter. Then here, what we'll be discussing in this topic, in this episode, introduction, cell cycle, and genetically controlled cell cycle, reasons for cell, why it divides or how it divides, mitogen and mitotic poison. Then after that, famous checkpoints. Checkpoint 1, main checkpoint, major checkpoint, restriction point, then minor checkpoint G2 to M. Then cell cycle, I phase and M phase, 2 areas. Then most of the time that will be taken for I phase. After that, M phase, then interphase, G1 as G2, each item, what all events will be taking place, G1, S, G2. Then connected with the G1, there is a deviation G0 phase or quiescent phase. Again, what all events will be in G0 phase, remain metabolically active but no longer proliferate. Then after that, S phase, DNA duplicates, invisible phase. Then S phase, we can find centriole divides in animal side, DNA replication in the nucleus. Then G2 phase, mainly tubulin synthesis, DNA synthesis stops. Then after that, 5 theory question, 5 NEET model question with the key. If you like this episode, subscribe or like it, share it, comment on it. We can meet later with another episode. Thank you.